I'm Zoe Grant, and I'm a new postdoc in the Bruneau lab, and I'm excited to introduce our next speaker, who is Dr. Pedro Miura, who'll be talking about coordinated alternative splicing and alternative polyadenylation. Dr. Miura obtained his PhD from the University of Ottawa in 2010 and undertook a postdoc at the Memorial Sloan Kettering Cancer Center in New York. He is currently an associate professor at the University of Nevada in Reno, where he has been since 2014. He was supported in his transition to junior faculty through the NIH-funded COBRE program for underfunded universities, and it was through this program that his connection with Benoit was strengthened, with Benoit mentoring Pedro through this program. And I've heard that this was a mutually successful mentorship, with Pedro successfully obtaining tenure and funding for his work, and Benoit gaining a new RA, Kevin So. And I think it's clearly a meaningful relationship for Benoit to see Dr. Miura speak today. So thank you so much for your time.
Thanks for that introduction, Zoe. Can you guys hear me okay? Great. Yeah, it's really an honor to be part of this symposium. My story with Benoit actually goes back even earlier. So I met him when I was in high school. I was working in the DeBold lab at the University of Ottawa, and just met him once. He came back from Harvard to give a talk at Ottawa, and I was really struck by him even way back then. He was the best dressed person in the room, as he is the best dressed person right now, I think, even way back then. So I'll start my talk and save roasting for later, I think. So thank you again. It's a little bit different type of talk I'm going to give today, but I hope you'll find it interesting. So again, thank you, Benoit. Benoit, he was kind enough to visit us in Reno several times, and I got to come to Gladstone and give a talk and participate in his lab meetings. So I'm really grateful for this relationship. So I study gene regulation and the processing of genes at the RNA level. So most of you are familiar with alternative splicing. The simplest type of alternative splicing is cassette exon alternative splicing. So it can remove or add in exons within the gene body. And the other thing we study is alternative polyadenylation. And so this is a level of RNA processing that happens at the 3' end, which can lead to different length 3' UTRs. What I want to talk to you today is how we've come upon this finding that these two RNA biogenesis events that are thought to be separate are actually can be very intricately linked. And so we call this coordinated alternative splicing and alternative polyadenylation. So alternative polyadenylation is highly prominent. It happens in more than 70% of human genes. We're interested in the type of alternative polyadenylation that happens at the 3' end within the 3' UTR. So this can lead to short UTRs versus long 3' UTRs for a given gene. We found when I was a postdoc in Eric Lai's lab that these longer versions tend to be more highly expressed in the nervous system. And this is the case from Drosophila all the way to humans. And we can see this type of change in APA, so we call it lengthening of 3' UTRs, in short-read RNA-seq data from Drosophila. Early embryos lack nervous system, late embryos contain a nervous system. So from this track shown at the DSCAM1 gene, you see that the signal for the RNA-seq drops off here in early embryos with no nervous system, but in later embryos, the pileup of reads here is indicative of increased expression of a longer 3' UTR isoform. And then in central nervous system samples, this long isoform predominates. We were interested in finding out what are the functions of long 3' UTR isoforms. There's thousands of them in mice, there's hundreds of them in Drosophila. And to do this, we took an approach in both in mouse and in Drosophila to knock them down using siRNA approaches, or to do CRISPR-Cas9 to delete the distal polyase sites such that the gene can only make a short version. We did this for the DSCAM1 gene for various reasons. And showing you first here an shRNA knockdown using this Gal4 UAS system, so we can specifically knock down in neurons. Doing this results in a loss of the long version, of DSCAM, but keeps the short version completely intact. So the knockdown works. We expected to see a recapitulation, at least partially, of what a DSCAM1 null mutant would look like, and that's what we found. We found that axon guidance, as expected, was impaired when we lost just this long version. These flies were severely impaired. They died by 10 days. These flies are not being anesthetized. They're just kind of hanging out. So severe locomotor defects in these animals. So we had previously conceived ideas of why a long 3' UTR version of a gene would be important. And this is because we know that 3' UTRs can be important for mRNA localization. They can be important for local translation of the mRNA. And this is thought to occur mostly through the presence of microRNA target sites and target sites for RNA binding proteins within 3' UTRs. So if we lose that long 3' UTR, we lose regulatory control, and we expect to see an impact on translation of that DSCAM1 gene. But what we found, curiously, was that even if we knock down that long version, the protein levels of DSCAM1 remained unchanged. So severe phenotype, but no change of that DSCAM1 overall protein levels. So we scratched our head a little bit and resorted to what I like to do, which is look at RNA-seq tracks, and we kind of found a potential answer. When we look at the DSCAM1 gene, it's highly alternatively spliced. In fact, it's the most alternatively spliced gene in nature, including humans. And we looked upstream of our interest region to what kind of changes were associated in early delayed embryos of these upstream exons. And we found that these two exons, exons 19 and 23, were showing changes associated with the development of the nervous system. We browsed this RNA-seq data and found a slight correlation that could be visualized with this older type of RNA-seq data that when this long UTR showed up, well, it seemed like these exons became less expressed. So more exon skipping when this long version appeared. So I started to think, maybe these things are connected. Maybe when we lose DSCAM1, the long version, we're also losing these specific alternatively spliced isoforms, changes in the protein coding region. As embryos develop, the long version appears. And in fact, yes, these two exons, exons 19 and 23, get skipped. So exon skipping is associated with the 3' UTR lengthening of this gene. Now, in order to figure out whether they were actually connected, we took a variety of approaches. I'll just show you one, which I think makes the point quite clearly. With the shRNA knockdown, I already showed you how we can knock down just the long version, but we hypothesized, well, if we could knock down the skipped version of this exon 19, so SH to the -19 version versus SH of the +19 version, then we think if this is actually connected, then loss of this -19 or targeting this -19 version would also result in loss of the long UTR. And that's what we got. When we knock down with the long, for the extended version, we lose the long UTR, but also when we knock down with this SH -19 shRNA, we also lose the long version. So the skipped exon 19 mRNA and the long mRNA are one and the same. So this left us with a few questions, many questions actually, and it's been really fun because we didn't expect at all to be studying alternative splicing. So really, the main thing we're interested in is, what are the mechanisms that could cause this? How is it just coincidence that the alternative splicing change and the APA changes happen during embryonic development, or is there something more? Secondly, what other genes are controlled by this type of coordinated splicing and polyadenylation? So we're working on this first one, and it's a little bit too early to give you too many clues about where we are on that. But I'd like to show you our progress on how we found ways to look at the other genes that are controlled in this fashion. Here's an example of one of these other genes, and it's called Tuttle. And I think it's a nice track because we can show you the entire gene body throughout embryonic development. And this is very old RNA-seq data, short-read RNA-seq. But it's quite evident that when this long UTR appears, we start to get also expression of this alternatively spliced isoform. And in this case, unlike the dsCAM version, which was skipping, this one's including. So including an exon when we have a long 3' UTR expressed. So we thought, how can we detect likely events that are coordinated at this alternative splicing and APA levels using existing data? And we were able to do this using conventional tools that are publicly available in order to quantify the change of APA and the change of alternative splicing within published RNA-seq data sets. In this example title, we can quantify the increase of the long 3' UTR here with this DPAU metric, and the PSI value for the cassette exon alternative splicing. We performed this analysis comparing early and late stage embryos and found around 58 genes in Drosophila that exhibit both in a coordinated fashion. So a significant change in 3' UTR lengthening and a significant change in exon skipping. And so the tools we used are publicly available, kappa and RMATs. Now we'd like to confirm these events, that it's not just coincidence that these two things happen at the same time, but that they're actually connected. And so we took this approach also in the dsCAM1 paper that I'm just showing you here for another gene called KHE73. And so this is using the Oxford nanopore MinION system, which some of you are familiar with. It's really, when these first came out, it was just so exciting, right? This little sequencer in your hand. And I'm still pretty fascinated the fact that we just plug it into a laptop. So we took one strategy, which is really just a long read sequencing of an RT-PCR product when you simplify it down to that. And we would do amplification of either a universal primer set that's going to amplify cDNA of this KHE73 gene within a certain region, but will detect both short and long versions of the transcript. And then it'll tell us what's the usage inside. And here's the data for that. You see that there's this alternative exon is used with inclusion rate of 33%. Then we do a second PCR and a second library with a long specific primer set. And so this will amplify only long 3' UTR containing transcripts from this region. And then it's evident that there's almost 100% inclusion of this alternative exon. So for this gene, like dsCAM1, there's a particular exon upstream that's connected to the use of a long 3' UTR. Now, there's some caveats to this approach because it relies on a lot of PCR amplification, and we can't get really information on exact connectivity of that short 3' UTR version. So we, Bongmin Bei and Jiping Zhang in the lab developed a method in which we pull down cDNA that's from embryos or from any RNA sample, using oligonucleotide probes that are complementary to common exons of the genes of interest. So for instance, in this first bunch that I'm going to show you, there's about 14 genes that we targeted, and with 3 to 5 of these oligonucleotide probes per gene. And then we perform nanopore sequencing using the MinION system. So here's just the raw data for this gene KHE73, which I showed you earlier. The alternative exon is right here. This is what we get when we throw it in the nanopore. Okay, so we get good coverage. This shelf, this step you see here is indicating where the short 3' UTR ends and the long 3' UTR proceeds. And what Jiping did was she parsed out the reads, these long reads, to say, well, I want to just look at what happens up here for the reads that are long, the reads that span this extended region. And this is what they look like. Here's the reads, and then you see how this exon and this exon both show a whole much more inclusion than what you see for the short only reads. So the short only reads, there's very little inclusion. This technique works really well. We're really happy with it. Here's another example, just simplified for the KHE73 gene, the unfiltered and then filtered for long, filtered for short. And it's just so obvious that these exons are really tightly connected to either long or short 3' UTRs. Works on our dsCAM1 gene, which we published on before. This is showing that exon 23, long UTR skips, short UTR includes. So This coordination of alternative splicing and alternative polynylation, we think it happens in anything with the nervous system. So actually, I'm not sure if Ben was aware, but one time in our meetings during these mentoring sessions, he suggested, oh, why don't you think about getting into mouse ES cells? Because we were already doing a lot of work with mice. And I let this idea sit for about 2 years, and now we've in the past year we started it up, and with the help with some reagents and expertise from Benoit and Elfej, so we're really grateful for that. And so Min in the lab, Bong Min, is able to now differentiate into glutamatergic neurons, and she did some analysis the same on some published data as same as the early embryo to late embryo. And we can see events that happen with alternative splicing and events that happen with APA. So thousands of genes lengthen when you go from ES cells to neurons. And we can get the overlap. So about 144 genes from this short read RNA sequencing analysis overlap. And so can we confirm that these are connected? And so Min's just begun doing this, and I'm gonna gloss over this just because kind of getting tight on time, but here's an example of this gene called ENDO-V. And in this case, using the pull down sequencing approach, we can get the entire gene from 5' end all the way to 3' end. And you see the reads dropping off here at the short 3' UTR. And then if you pull just the long ones, which aren't even visible, they're such a small proportion relatively, we can pull out long 3' UTR specific transcripts. And you can notice that there's a difference in this alternative splicing event upstream. So it seems like this technique works quite well. It's not very complicated, even though it's not obvious to get to work either. But we would like to quantify these events genome wide. So the take home message here is that long 3' UTR selection can be linked to upstream alternative splicing events. We're looking at cassette exon alternative splicing, but we have good reason to think that this can link to other biogenesis events such as alternative promoter selection. Our approach of using cDNA pull down followed by long read sequencing can quantify these coordinated events. And what I didn't get to again was the mechanism. We have data that suggests, and it's in this 2019 paper, that the first thing people would think is that maybe this event here, because it happens first, it's going to influence this event here. And actually, we have evidence that the opposite is happening. That this 3' UTR is actually required for proper splicing that happens upstream, temporally earlier than the cleavage and polyadenylation. So with that, a lot of talented people in the lab that are moving on to do great things. So as Zoe mentioned, Kevin was a student in my lab as an undergrad, and then moved on to work as a research associate with Benoit, had a great experience, and now is a PhD student at Harvard. Lauren Eggleston worked directly on this work of the coordinated APA, and Bong Min and Ji Ping have been are really leading this project now on this coordinated APA. So thanks to collaborators, mentoring, and I'm happy to take any questions.
Great, thank you so much, Dr. Miura, for that really interesting talk. If anyone has a question, feel free to type it into the chat. If not, I'll ask about the striking phenotype that you have in the DS-CAM flies, and I wondered how the alternative splicing event, what's the mechanism for leading to that cellular phenotype?
Yeah, so it's kind of a trick question because you lose both the long UTR version of this transcript, and you also lose the exon. So which one's more important? Ideally, we would do some kind of rescue experiment where you get rid of the long UTR and the splicing event, and then you rescue just with the right cassette exon protein coding region, or you rescue just with the 3' UTR. So we can't really, that doesn't work too well because of dosage. So we think both are important. But my money would be on, just because the phenotype's so severe, my money would be on the actual protein coding region being the important part. So we don't know exactly there's some phosphorylation sites in that alternative exon, so that might play a role. But really what this is, it's a new way of thinking of how a 3' UTR can influence gene expression, because it can actually change what type of protein is made, even though that event is very far away from the protein coding region of interest.